Hello, and welcome to part 2 of this series on DNA variant interpretation. I'm Sam Strome, PhD, Assistant Professor of Pathology and Laboratory Medicine, UCLA. Today we're going to go over two of the most essential concepts in DNA variant interpretation, allele frequency and variant classification. As I mentioned in part 1 of this series, the average person has about 4 to 10 million differences between their genome and the human genome reference sequence. Only a fraction of these variants are located within parts of the genome encoding proteins, which we collectively call the exome. And only a fraction of exome variants are located within the 2,000 to 3,000 genes suspected or known to be related to the disease. Looking at only those variants which are located within a known disease gene still only narrows it down to about 6,000 variants per person. Let's look at an example variant to get an idea of what allele frequency really means. This is a real variant, almost 38 million bases from the start of chromosome 4. According to the Human Genome Reference Sequence, the expected nucleotide at this position is A. The allele frequency of this DNA variant is defined as the proportion of chromosomes in a population that has the G allele at this position. To figure out the allele frequency, I've collected 50 smiling green people to help participate in my study. They may not be smiling for long though, because I'm going to take a small sample of blood. Using molecular techniques such as DNA sequencing, I'm going to determine which alleles are present in which individual. This is called genotyping, because each person has 2 chromosomes, one from mom, one from dad. There are 3 possibilities: homozygous reference, AA, heterozygous, AG, or homozygous, GG. The results of my genotyping experiments have revealed that 4 of these 50 people are heterozygous AG, and 1 of them is homozygous GG. The remaining 44 people are homozygous reference AA. The minor allele frequency is calculated as the percentage of chromosomes in a population that carries the variant. To do this, you add the number of heterozygotes to 2 times the number of homozygotes and divide by the number of chromosomes. In our example, this works out to a minor allele frequency of 6%. So why is allele frequency so important? Because population genetics tells us what frequencies to expect for various modes of inheritance. Let's back up for a second and go over some population genetics basics. Hopefully, you remember from high school or college the idea of Hardy-Weinberg equilibrium. It's a way to calculate the expected rates of homozygotes and heterozygotes for an allele based on the variant frequency alone. Let's consider our variant with the allele frequency 6%. In Hardy-Weinberg, we'd say that Q equals 0.06. P is always 1 minus Q, so P is 0.96. When we do the math, over 90% should be homozygous for the reference allele. That's pretty much as you'd expect, right? When we calculate 2pq, we find that over 11% of people should be heterozygous. If this variant causes an autosomal dominant condition, we would expect that 1 in 10 people would have that condition. That means it's really far too common to cause a rare dominant disease. By calculating p squared, we find that less than 1% of people should be homozygous for this variant. That may seem rare. After all, only 1 out of 278 people would be homozygous. However, that's still too common to cause a rare genetic condition. Let's go back to this table. Hopefully it makes a little more sense now. In the middle column, I've listed the Hardy-Weinberg style genotypes consistent with the listed modes of inheritance. In the maximum frequency column, I've listed the highest possible allele frequency that could lead to a disease as rare as 1 per 10,000 individuals, which is consistent with most rare genetic conditions. So if we want to make any sense out of the 6,000 or so gene-altering variants expected per person, we can use our knowledge of allele frequency to focus on new or very rare variants. Lucky for us, due to some population math I'm not going to go into here, the majority of variants observed in any single person are common. On this chart, the blue line rises from left to right, indicating that the rarest variants are the least common type in any one person. If you look across a population, however, the largest proportion of variants is the rare variants. This is because when you are searching for new variants in a population, observing a common variant in 10 different people only counts once. Every time you sequence a new person, you get a handful of new variants, Across thousands of people, that really adds up. We'd love to know the allele frequency of all possible variants at all possible locations. After all, there are only about 9 billion different single nucleotide changes, if you think about it. The good news is that much of this work has already been accomplished. Groups like the International HapMap Project, the 1000 Genomes Project, and the Exome Sequencing Project have sequenced a pretty big number of people and released their results to the public. Much of this information is stored in the Database of Short Genetic Variants, or DBSNP for short. We're not all the way there yet, though. There is still a long way to go before enough people from enough different places have been sequenced. For now, we can use the data available to narrow down the list of possible disease-causing variants to fewer than 200 per exome, on average. After allele frequency, the next most important aspect of DNA variant interpretation is function. Variant function can be broken down into two main categories: the change in the amino acid sequence and the impact that change has on the biology of cells. The change in the protein amino acid sequence resulting from a DNA variant can usually be easily predicted using the genetic code. The impact a change has on the biology of cells can only be understood by laboratory research. Because lab research is costly, time-consuming, and not always feasible, it is the exception and not the rule when the cell biology of a variant is well understood. The genetic code is the relationship between DNA and proteins. Each 3-base codon of a messenger RNA is code for one of the 20 amino acids, or marks the end of the protein sequence, what we call a termination codon. Let's consider this fictitious mRNA sequence. At ribosomes, the cell's protein factory, the mRNA is paired with translational RNAs, or tRNAs. tRNAs are amazing molecules. They're the physical embodiment of the genetic code. They have a complementary RNA codon at one end, and the corresponding amino acid attached to the other end. The ribosome grows a new protein by connecting the amino acids on the end of neighboring tRNAs. This process continues until the termination codon is reached. The tRNA corresponding to a stop codon simply has no amino acid attached, so the chain cannot be continued. All genes that code for proteins begin with an AUG codon, which encodes the amino acid methionine, and end with the termination codon, which encodes for no amino acid at all. Because there are more 3-letter combinations than amino acids, some DNA variants have no impact on the protein sequence of its gene. In this example, the 9th coding base of this mRNA is changed from an A to a G, so the coding changed is named C.9A to G. The leucine amino acid at this position is not changed, so the protein change is P.LEU3LEU. This type of variant is called synonymous. Because they do not directly alter the primary protein sequence of a gene, these variants are often considered unlikely to be disease-associated, though there are exceptions. A missense variant is one that alters a single amino acid. In this example, the C at coding position 10 is changed to an A, resulting in a change from a leucine to isoleucine at amino acid position 4. Missense variants can be difficult to interpret because the range of possible functional effects they may cause is very broad. A nonsense variant is one that results in an early termination codon. In this example, the T at coding position 8 of the mRNA has been changed to an A, resulting in a stop codon at amino acid position 3. This type of variant is very likely to result in little or no functional protein from that allele. A frame shift is an insertion or deletion variant that is not a multiple of 3. In this example, a single base insertion of a C. With this type of variant, all subsequent codons are affected. In this example, the variant peptide is completely different and keeps right on going past the original stop codon. This allele will likely produce a garbled protein of no use at all to the cell. Most eukaryotic genes, including those in mammals like we humans, include both exons and introns. Introns do not encode for proteins, but are transcribed onto the initial mRNA. Splicing is the process of removing those introns from the mRNA, leaving only the exons. The DNA sequences at the boundaries between introns and exons are not flexible. In humans, the first and last two bases of introns and exons have to match the pattern pictured here, or no splice will occur. These positions are called the canonical splice sites. Variants at canonical splice sites lead to the loss of an entire exon or the inclusion of an extra sequence, acting like a large insertion or deletion. These are typically considered highly likely to damage the protein product, with some exceptions. In this example, the first base of an intron, originally a C, is changed to an A. This results in a loss of splicing initiation, so the mRNA now includes intronic material. This could be bad news for the protein, as intronic sequences do not have protein coding information in them. Because it's not possible to reliably predict the protein product generated by an allele with a canonical splice site variant, the accepted way to name this protein change is P.?. In this table, I've listed the major classes of DNA variants and some information about them. In general, the variants that wipe out an entire protein are the most suspicious. However, all of these types of variants have been associated with multiple inherited diseases. Thanks for watching part 2 of this series. I hope it was helpful. Please leave questions or comments here on YouTube, or send them to me at Twitter @StromDNA.